The overall goal of this procedure is to measure attachment and internalization of influenza A virus particles into A549 lung epithelial cells. This is accomplished by first cold binding biatenylated influenza A virus to A549 cells, which allows attachment but prevents uptake of viral particles. Next, the virus is allowed to internalize by shifting the temperature to 37 degrees Celsius. Then, a blocking step in which unlabeled streptavidin is bound to cell surface attached viruses is performed in order to discriminate between attached and internalized viral particles. Finally, the cell-associated biatenylated virus is incubated with Cy3 labeled streptavidin, and flow cytometry is used to detect the stained viruses. Ultimately, the relative amount of attached and internalized viruses is calculated.
Though this method can provide insight into the relative amount of influenza A virus that attaches and internalizes into A549 cells, it can also be applied to other systems using different cell lines or other viruses.
After generating biatenylated virus and determining the required amounts of the labeled virus, streptavidin, and Cy3 labeled streptavidin according to the text protocol, prepare 4 deep well tubes, each containing 200,000 A549 cells. Label the tubes as 0 minutes, 0 minutes plus STV, 30 minutes, and 30 minutes plus STV. Next, spin the tubes, remove the supernatant,